Good afternoon, dear webinar participants. I want to give you a little overview about protein detection techniques in electrophoresis gels. And you see already on the first slide that there are several, and I have a list of them, and we will discuss them one after the other. So first of all, it is possible to apply a negative staining, which stains only the gel but not the bands or the spots. This has long time been the preferred technique by people who have to do downstream analysis like blotting and mass spectrometry. But the most applied staining technique, I would say, is Coomassie blue staining. It was the first on, or almost the first for polyacrylamide gels. And there are different versions of Coomassie blue staining, colloidal, monodisperse stainings, and I will introduce a few of them and talk about what is better, what is not so good. Another very frequently used detection technique is silver staining, because silver staining is very, very sensitive. On the other hand, it's quite cumbersome to do, a lot of staining of steps, and is not useful for quantification. I will come back on this matter later on when we talk about silver staining. In the last couple of years, fluorescent staining has become quite interesting. A lot of people have started to apply this method because it has a lot of benefits for very sensitive and quantitative detection, because there's a wide dynamic range. I will also talk about a technique which is very, very rarely used in 2D electrophoresis, but nevertheless I want to explain why we tried it and that it works. And the last thing I will talk about is fluorescent labeling. Fluorescent labeling is on the market since more than 11 years already, mostly for the technology called DIGE, difference gel electrophoresis, where you can label with different dyes and run the multiplex the samples in one gel. But I will also introduce a technique for fluorescent labeling as a replacement for normal staining, just for one sample. Okay, I will start with the negative staining. Just a moment. Yeah, here we go. There is a, before I start with the negative staining, there is an overview over the techniques. So we start with the Coomassie staining because it's the mostly used one, and I list all the others for sensitivity limits, quantification properties. Do you need living cells for labeling or not, and what is the dynamic range? Now, Coomassie blue staining is really used a lot because there is one benefit: it's not expensive, and you can. The second benefit is you can directly see the result after a relatively short time, and it's within some limits quite a quantitative technique. The sensitivity limit, which is shown here, says 20 nanograms to 5 nanograms. Yeah, this is dependent on the type of Coomassie blue staining you are using. So there are different staining techniques give you different sensitivities, but also the proteins themselves, they bind the dye differently. So for instance, albumin binds a lot more dye than other proteins, collagen molecules, for instance. Yeah, that has to be bear in mind. Negative staining is also quite sensitive, but the trouble here is you cannot use it for quantitative techniques. It does not modify the proteins. That's why a lot of people like it for blotting and for and also for mass spectrometry, but for quantification not easy, and also for visualization. I will show you. You will see that in the next slide. Silver staining, sensitivity limit down to about 200 or a little bit less picogram, also depends on the modification of silver staining. There are several around. I will introduce two of them to you. But unfortunately, silver staining is not useful for quantification. Of course, you will see some papers, publications where people try to read out some quantitative results from silver stain results, but I would really not rely on that, because silver staining is not really a staining technique, it's a complexing technique. What you see is metallic silver complexes with proteins, or with DNA also. It stains also, it detects also DNA. Fluorescent staining is very sensitive, you could say almost as sensitive as silver staining. Very good in quantification because these fluorescent dyes have a wide dynamic range. Disadvantage of all these fluorescent techniques, unfortunately, is that you need a fluorescent scanner or fluorescent camera system. You cannot see the result directly, and fluorescent cameras and scanners are not so really cheap. Another technique started to introduce already is fluorescent labeling. Fluorescent labeling can be made very, very sensitive. For instance, with saturation labeling of cysteines, there you can come very deeply down in the sensitivity. Is also good for sensitivity, for quantification, like fluorescent staining. And I will show you a new dye in the end of this presentation. Very sensitive and very valuable for quantification are the radioactivity labeling or radioactivity detection techniques, which use X-ray film or phosphor imager plates for detection. But there are less and less people use them because of the troubles with radioactivity in the lab. And finally, just to make the list complete, there is stable isotope labeling, but here the detection is done with a mass spectrometer, and you can also do multiplexing by using differently heavy isotopes. And but this will not be covered during this webinar. Yeah, here you see this is called SERVA Snow. So you can, of course, put together your reagents yourself and make your staining yourself. But for the convenience, you can also order that as a ready product, and you see immediately in the picture why we call it SERVA Snow. It looks like 2-3 weeks in advance in the pre-Christmas time when we have these white dots. Yeah, normally you see it in the reverse way. You see a white background and dark spots when you photograph it against a dark black background. But here in this way of looking at it, you see the spots a little bit better. And you can understand you cannot quantitate the proteins because you stain the background and not the proteins themselves. So it's based on Imidazole-Zinc, and you can also use it as a reverse stain. So if you want to blot, for instance, transfer the proteins through an electric field, you can just, with an EDTA solution, remove the metallochelates, and then the proteins become mobile again. You can transfer the proteins onto a blotting membrane. Here you see the first example of a protein staining technique, which is Coomassie R-250. You may be aware that there are, of the Coomassie blue, there are two types of Coomassie blue. There is R-250 and G-250. This here is R because it has a reddish characters, more red-blue, what you see here. And this was a monodisperse solution, so you are staining in presence of methanol and acetic acid, and then you have to destain the background. We offer also this as a kit that's very easy to use, easier to set up. You don't need to weigh in the Coomassie dye and but you can, of course, also buy Coomassie powder and make your own solutions. Sensitivity is quite nice, and you see the background is also quite low, and it's in some limits, as I said, you can use it for quantification of the proteins. That is a typical image of Coomassie G-250. This is a colloidal staining. What you see here, you can use Coomassie G-250 for both, for monodisperse and also colloidal staining. But colloidal staining can only be performed with G-250. So this more greenish appearance of the dye, and the difference between G and R-250 is that the G-250 has two additional methyl groups, which the R does not have, and makes also the dye more hydrophobic. It's also used for so-called blue native electrophoresis because it's more hydrophobic dye and binds to hydrophobic surfaces of complexes and proteins. So people who have really tried a lot of different staining techniques claim that there is a Coomassie G-250 colloidal staining protocol which is the most sensitive among all the others, and it's coming close to silver staining. And this one, what we hear, should be the most sensitive technique. We have also converted into a kit, so you can make it yourself. You can read the protocol for it, the recipe, but you also can order it as a ready product, and then it's quite simple to do. What you do here is usually you fix the proteins for a short while, and you wash the gel, and then you submerge the gel for about 2-3 hours in the colloidal Coomassie dye. You will have a complete endpoint staining method with this, and destaining here is performed just with water, just with pure water overnight, and yeah, works very well, very easy to do. Now here you see a mass spec, MS means mass spec compatible silver staining. This was E. coli extract separation in a thin horizontal gel on a film support, and there are people who prefer silver staining over Coomassie staining for mass spec analysis because they claim they get cleaner material after digestion. What is very important to make silver staining mass spec compatible is that you omit glutaraldehyde and other cross-linking steps, because once you have cross-linked your proteins in the polyacrylamide gel, also in-gel digestion does not help you anymore. You can't get your proteins or your, I would say, your peptides out of the gel anymore. It's not as sensitive as a non-mass spec compatible silver stain, but when you have optimized the protocol, it's quite good. Also, this kit is available from us, an optimized procedure. I would estimate there are about 10 different mass spec compatible silver staining protocols around, and we have tried them all, and we have selected the most sensitive and the best signal to noise protocol. You see one result here. Now, for some types of electrophoresis, like for isoelectric focusing, this sensitivity of this previous silver staining is not sufficient. The previous silver staining, I go back to the slide before, is based on silver nitrate incubation, and then the development is done under basic conditions, whereas this so-called ammoniacal silver staining is developed under acidic conditions. You have an ammoniacalic silver incubation. This technique is very, very sensitive and is mostly used for isoelectric focusing, particularly when you have very basic proteins to detect. Here, this is an example, a focus gel pH 6 to 11, so 6 to 11 means pH 6 to 11 pH gradient with 24 slots, so these are the slots for sample application. And this multi-step silver staining procedure gives you the highest sensitivity. You can, of course, also apply it on 2D gels, but it's very difficult to make this, or maybe impossible to make this silver staining modification mass spec compatible. You should also note that you need about 10 times as much silver concentration for silvering than for the other technique. But it's routinely used, at least in Germany, in the neurology labs. The blue box you see here is the latest version of a camera system that we have, where you can take pictures of your gels for visible and also with UV detection. We also have a scanner, but this scanner is only available for visible detection that we have. It can be used in transmission and reflection mode. Transmission mode is very important for quantification, but you can only apply it for films, for silver staining, for Coomassie, for the bands that you are seeing, or spots that you are seeing, not for fluorescent. It has a very good dynamic range. The one with the 3.7 OD is the Bio 5000 Plus. The older version had over 4 ODs. It's a 16-bit detection, and what you see here, the drawer, this is a glass holder where you can place the wet gels or the dry gels into it. From my experience, I have worked with many different scanners. I must say this is the best scanner I've used in my life, so to say. Okay, but only for visible bands or spots. Now I want to point on a second problem with silver staining. The first problem is that silver staining cannot be used for quantification because it's not an attachment of a dye or binding of a dye to a protein moieties, which is quantitative. It's just forming complexes with the proteins or with DNA molecules in a gel. Some people even call silver staining a disturbance in a polyacrylamide gel. What you see here, there was the same sample, E. coli extract, in the same amount, 20 micrograms, applied on 2 IPG strips and then run in 2 mini gels, one with 12% T and one with 14% T, and it has been stained and developed by the same person in our lab. And what it shows you is it's very difficult to find the right moment to stop the development. If you stop the development too late, you get a too high background, and some of the spots are merging into each other. If you stop too early, then you miss a lot of the spots. The problem is that silver staining is not an endpoint method. That is the second reason why quantification with silver staining is practically impossible. Now, this is a criticism on silver staining. We have marketed since several years already a dye which was called SERVA Purple, which was the same like Deep Purple marketed by company GE. But then the production team who produced the dye could not maintain the production as it used to be before, and then we had to look for a new source, and we found a producer who is even better, a synthetic dye, which is the same molecule like the biologically produced lava or Deep Purple. We call it SERVA Purple, so this is very new, and it's also very new news to you that we have that in our portfolio. And if you test it against the old dye or the former dye, you will see that the performance is better, that the sensitivity is higher, and so it's very nice, very well quantifiable dye. Here you see a 1D separation, markers, HeLa cell extracts, and E. coli extract, and it's very useful for quantification. Oh, I repeat myself, sorry for that. Here you see application of this dye in a 2D electrophoresis gel, again E. coli extract in a pH gradient 5 to 8, and you see the performance is very well and very good, and no speckles, and very good sensitivity. Now to the next. You might not be familiar with fluorescent intermediate staining. What is this? And why would we try fluorescent intermediate staining? Now, all the post-staining methods need work after the separation. You have to fix the spots, or the bands, you have to fix the spots, you have to stain them, de-stain them. So this requires several steps in a certain while after the separation until you can see the result. Of course, the pre-labeling techniques like Deitsch is much easier because you can directly take the gel and put it under a camera system or on a scanner and have the result. And you don't need to fix the proteins. You can directly afterwards transfer electrophoretically the proteins onto a blotting membrane because the protein bands and spots have not been fixed. Another thing which disturbed us a little bit was when you use these standard dyes, these Psi dyes on the market, which are binding to lysine or cysteines, you cannot have carrier ampholites, or you must not have carrier ampholites and reductants in the sample solution because they interfere with the labeling. Now there are samples around which are a little bit tricky to extract and which are not so very soluble. For them, it's very useful to have carrier ampholites and reductants in the sample solution. So we thought it was a good idea to first use a standard sample preparation technique, run the samples on the IPG strips in the first dimension, and then in between the first and the second dimension, during the equilibration of the strips with DTT and iodoacetamide, during this step you apply also the dye, the label for the proteins. So you are not restricted in the additives for the sample preparation, and you can directly scan the result afterwards. So we have tried it, and we were very surprised how well it worked with the dye. I introduce here to you the SERVA HPE Lightning Red, a fluorescent dye, which has the same same exciting wavelengths, 530, so green light for excitation, and emission wavelengths maximum is about 610, and this is about 610. So the standard settings can be used. Now this works very well, and what you see immediately with the same sample, you don't see any modifications of the running distance in STS electrophoresis. That was a very interesting result, and also when you applied more dye than usual, you do not see any over-staining effects, and the samples, the proteins, did not become more hydrophobic. That made us very brave, and so we tried to use this dye also for pre-labeling the sample directly before the sample application. And we tried that first with 1D electrophoresis, and here you see 4 mini gels where the same samples and the same amount of these samples have been loaded, and they have been detected with monodispersal Coomassie blue first, then with silver staining. And what you see immediately in silver staining, the number 7 lane is highly overstained, so we stopped the development. In this way, we could not get the full sensitivity for the markers, but on the other hand, you cannot see any single band here anymore. On the left down here, you see SERVA Purple. This is very nice post-staining fluorescent technique. And here with SERVA Lightning Red we have higher sensitivity and very good quantitative results. Just here I want to show you an overview of a protein detection workflows. And here you see immediately the difference between labeling and staining. All the staining techniques need longer and more or less staining, fixing, staining and destaining steps. And sure, there are different methods for Coomassie blue, as I mentioned already, monodisperse and colloidal, which are different also in the time consummation, or also there are different silver staining methods, but they all have in common that they have relatively many steps and relatively long detection times. The fluorescent stainings, including SERVA Purple, also need a fixing, staining, washing step until you can image them. And the unique thing with a label is that you label first for 30 minutes and then you can directly image. Or what you also can do, you can fix the image, the gel before imaging, or you can fix afterwards, or not fix at all. You can directly transfer the proteins from the gel onto a blotting membrane. So you have a lot of possibilities. Now what you see here is a 2D gel. Yes, it was one gel, it's the identical gel, where we had labeled the sample with our SERVA HPE Lightning Red before, and then after the separation, in this case we have fixed the gels and these protein spots with 10% acetic acid and 15%, 20% methanol, and then we have scanned the image, and afterwards then we have silver stained the same gel. Now when you compare the two results, everybody at the first glance would say, oh, I would say the silver stained gel is better. You have really nice sharp spots. They are well to recognize, good to see, and the image of the lightning red is not as intense. But when you look a little bit longer on the two of them, you will realize that the silver stain image is more like a black and white, 100% black and 100% white image. And when you look even more thoroughly, you will see that all the spots that you see on the silver stain spots gel, you see also on the fluorescent scanned gels, but just with different intensities, and they give you the intensity differences which are necessary for quantification. And if you even would longer look at them, you would realize that there are a lot of spots in the fluorescent image which you don't even see in the silver stain image. So you find more proteins in the fluorescent detection than after the highly sensitive silver stain. Now we have scanned these gels, and we have taken the data to Delta 2D. This is a software, image analysis software from the company Decoder, and the result is shown here with false color representation. The green indicates lightning red spots. The red indicates the silver stain, and when you ask for a list for protein spots, it is seen that in a mini gel like this, in a 7x7 mini gel, you have already 20 more spots with the fluorescent detection than with the silver staining. Now we have, of course, applied this also to large gels. This is just one example on a large gels, on an HPE gel. And this here, just for the curiosity, I have to tell you, this has been fixed and transported in a car for more than a week, and then later on scanned again, and we did not see a difference between the first scan and the second scan. So fixing does not cause a reduction of the fluorescent signal. This is a very useful feature of this particular dye. And here you see another image of an HPE gel, but directly scanned after the separation. I want to list the advantages that we see with using the lightning red, SERVA lightning red. So you have the direct detection directly after the separation without any further steps. No staining, no washing steps, no fixing steps, so that makes life much, much easier. And it's also environmental friendly, I would say. We should not forget this point. You can, if you like, after imaging the gel, process with Western blotting because you don't need to fix the proteins. Sensitivity is very high. We believe, or we saw, even better than silver staining, less than 100 picogram protein per spot. We have a very wide dynamic range and a linear range of 10 orders of magnitude, which makes a very, very useful for quantification. We did not see any overstaining effects like they have become noted with side dyes. When you use too much of them for lysine labeling, the proteins become very hydrophobic and can precipitate out of the solution. And we also did not see a mass shift. We have used something more than minimal labeling. We have at least labeled with 10 times as much, and we did not see a mass shift. The labeling is unaffected by carrier amphilites and reductants. That is also a very nice feature because we are not limited with the sample preparation technique. It's just important that you have a pH above 7. That's important. I would say optimally about pH 8. It's fully mass spec compatible. That's another feature that we had tested by some of our customers who have used this dye already for labeling their proteins. Yeah, that is what I wanted to tell you, and I'm at the end of the webinar, and please, if you have any questions, I'm happy to answer them.